Good afternoon, everyone, and welcome to the Weight Research Institute seminar series. This afternoon, we have two of our EMCRs from the School of Ag, Food and Wine presenting to us. And the first speaker is Zhou from Matt Gillingham's group. Bo's been with Matt for quite a number of years now, and his talk is entitled GABA is a plant guard cell signal.
Hello everyone, my name is Zhou. I'm a postdoc from Matt Gillingham's lab, and today I will give a talk about my research. GABA is a plant guard cell signal. No. Okay. So gamma-aminobacteric acid, GABA, is a non-protein amino acid found both plant and animals. In animals, it's well known a neuron transmitter signals, and in plants, as a metabolite, it has been found to regulate growth and development. It's not clear that whether if plant use it to cope with stress, as it accumulates in plant tissue under stress, such as viral, salt, drought, cold, and mechanical damage. And that's why, in 2 decades ago, GABA has been questioned if it acts as plant signal. And however, no study has ever definitely proved this. In my research areas, as plant drought tolerance, so we first validate the GABA metabolism in the model plant Arabidopsis and the drought. We challenge the plant with the drought, and we find that comparing to the well water conditions, the available water content in leaves gradually reduced following drought treatments. And this is because the treatment rapidly reduced the water content in the soil, and that caused the water deficit stress, and the stress the plant associated with higher gamma accumulation in the leaves. So we wish to know why GABA is produced in leaves. And GABA is synthesized in the GABA shunt. It's converted from glutamate by glutamate decarboxylase. This process is also regulated by calcium because these GAT enzymes contain the calcium chymotrypsin binding domain. In Arabidopsis, there are 5 GAT isoforms, and GAT2 is the dominant one expressed in leaves. So we isolate the GAT2 TDA insertional mutant and find that those mutant plants reduced at least 75% GABA accumulation in leaves, but not in root. And under drought condition, the drought gradually built up GABA accumulation in wild type plants, but not in the mutant. Later, we find both mutant plant has the largest stomatal aperture when we measuring stomatal aperture width, which shown here, typical guard cell, and that's the red arrow point aperture width. And that doesn't change stomata density. So the largest stomata aperture, which means they will have more open stomata, and that's also here's the images, the typical stomata imagings from the plant, like the Arabidopsis over here. And that's the surrounded by pair of guard cells, and this is the aperture ball. So this is the gatekeeper of the gas exchange, CO2 uptake, and the water transpiration and oxygen release. So these mutant plants have more open stomata, they have a higher stomata conductance. The result of these is under drought or water deficit stress, the water content in the mutant plant will drop more rapidly compared to the control plant, the wild types, and that's make mutant plant more drought sensitive. So from these, we can see that the GAT2 gene encodes the major enzyme catalyzed GABA synthesis and drought. And GABA accumulation does seem has some role in regulation plant gas exchange. And we want to further prove that is where does GABA do this? So we did the GAT2 expression pattern analysis, and from this gas staining, we can see GAT2 highly expressed in whole leaves, particularly high in gut cell, which suggests that GABA possibly could be produced in the gut cell to regulate stomata movement. To prove this, we made cell-specific complementation on mutant plant. We used the gut cell-specific promoter called GC1 to drive an active form of GAT2 gene with truncation of promoting binding domain. When it tagged with GFP, we can see a highly and exclusively GFP fluorescence from gut cell. And then we examine the plant without GFP tag, and what we find is the complementation first recovered GABA accumulation, and also the high stomach aperture and high conductance also bring back to the wild type levels. And that makes the complementation plant had similar performance with wild type plant under drought stress, as we see the lower panel here. After 8 days stop watering, the GAT2 mutant is wilting, but not happen to the mutant plant. And let's go back to look at this image. That GAT2 highly expressed in whole leaf, not just in gut cell, also including some other cell type like mesophyll cells. This is consistent with previous research that investigates the global gene expression profile in both gut cell and mesophyll cells protoplast. And here the gene highlighted, GAT2, is highly detected in both cell types. So to know if GABA has any role in mesophyll cells, we used enhanced track line. It's called JR11.2. This line has the scale for UAS elements insert downstream of spongy mesophyll specific promoter. And this mesophyll promoter will drive the GAL4 expressed, further activated the UAS GFP expressed in mesophyll cells as shown in this image. So that's GFP expressed in the spongy mesophyll cells, which close to the lower epidermal layer with abundant stomata. So to complement the GAT2 back to the mesophyll cell, we just use this UAS element to drive the GAT2 gene. To do this, we first cross the GAT2 with JR11.2 to segregated GAT2 background JR11.2. Then we put the UAS GAT2 back to this plant. And we find the GAT2 background JR11.2 still has high stomatal conductance. And the mesophyll complementation, however, did not restore the phenotype. So take it as a brief summary, the GABA accumulation in gut cell controlled plant gas exchange. And this accumulation is essential for drought tolerance. And next step, we should how GABA make this happen. So this is so we presume that GABA can regulate stomata movement, which the gatekeeper of plant transpiration. And this stomata movement, either opening or closure, it responds to environment stimulus. For instance, here, when we move the plant from light to the dark, this can mimic the day to night transition. The dark would gradually close stomata. As you can see, reduce the aperture width from about 3 micron to 1. And in the opposite way, the light can stimulate stomata opening. And to prove if GABA has any effect on stomata movement, we did a very simple assay called epidermal strip assay. We first peeled epidermal strips, which contain stomata, incubate in a buffer under light. Then we add in GABA and put back to the light in the measuring stomata aperture width to see the status stomata. And however, we find no difference. And also we incubate under dark and adding GABA and keep it in the dark, and again find no difference. Here you can see the aperture width under dark is much smaller than the under light. That's because the dark closes stomata. So from these data, we can see the GABA at least does not stimulate stomach movement, either opening or closure. And we did another way, is we first incubate in the dark to close all stomata and adding the GABA and transfer to the light. We can see the light increases on the aperture, which means open the stomata. However, this was inhibited by GABA supplement. So which means the GABA probably is prevent stomach opening instead of promoting closure. And also we find it, we first incubate under light by adding GABA and put to the dark closed stomata, and again that's inhibited by GABA. So the overall is GABA more like this break of stomata movement. A similar kind of response to the GABA, we not just limit in the Arabidopsis, we see similar response in other plant species, such as here, the broad bean, the tobacco, soybean, and the barley. Because limit time here wouldn't list all details result here. And the take-home message is GABA is like a universal stomata modifier in different plant species. And apart from the epidermal strip assay, we also validate GABA effects in the intact leaves. So we cut the leaf from the plant and immerse the PTO into solution and the second part. And this solution is artificial xylem sap solutions. Then we use the ERGA, which the infrared gas analyzer like to record stomach conductance, transpiration rate, and the photosynthetic rate. And we can add some chemicals into the xylem solutions to apply treatment. And over here, you can see the gray area, that's dark treatment. And the white region, it's light treatment. So when the light down, we see a gradually increase of stomata conductance. That's because the light gradually opens stomata. And this, however, is reduced by GABA supplement. However, that doesn't affect photosynthesis rate, which means when we use photosynthesis rate to divide photosynthesis rate against stomata conductance or transpiration, we expect to tell transiently how much water used by plant to synthesize or to assimilate 1 unit of CO2. That would expect to tell the water use efficiency. And indeed here, GABA increases the water use efficiency. So that kind of helped understand why the GABA biosynthesis is increased on the drought. That was because probably can help to increase the plant's water use efficiency and save the water use. But also very interesting here that an epidermal strip assay, we see the GABA reduced stomata closure. But however, we didn't see clearly from intact leaves. So the reason for that is so far we're not quite sure, but we're assuming that the stomata aperture assay we performed on epidermal strip lack the mesophyll cells, which means it doesn't have the complete set of GABA cell signaling pathway. That may differentiate the stomata response on epidermal strips from intact leaves. Because of these, the later experiment we mainly focused on why GABA reduces stomata opening. Back to 2015, our group has been published a paper showing that GABA can negatively regulate a family of plant anion transporter. It's called aluminium activated mainlate transporter, LMT. Here is an example from wheat TALMT1. With increase of GABA treatment, it actually reduced the activity of the malate flux by these proteins. And in the Arabidopsis, there are a number of ARMT homologous identifier in the guard cell. And among them, the ARMT9 and the 12 are the most well-studied members. They respectively control stomata opening and closure. For instance, here, ARMT9, it targets on the tonoplasmic membrane to catalyze the anions uptake and accumulation in the guard cell vacuum. That will increase the turgor pressure of guard cell and cause a swelling of the guard cell and make stomata opened. And goes back to look at these data. In the wild type plants, when we apply the GABA, it reduced the stomata opening. That's reminiscent of what happened to the ARMT9 mutant, as shown over here. So that's really assuming this GABA has inhibitory effect on the ARMT9 to mimic the loss of the ARMT9 loss of function. To do that, we also get this ARMT9 mutant plant and do the same assay. We find the stomata opening process showed insensitive to GABA. And when we do recomplementation, ARMT9 back to this mutant and GABA sensitivity came back. So this signifies that strongly proves that ARMT9 probably the target by GABA during opening. And what's more is when we go back, look at the GAD2 background plant, low GABA accumulation, high stomata conductance, and we further knock out ARMT9 to generate double GAD2 ARMT9 double mutant. Again, has low GABA accumulation, but stomata conductance brought back to the wild type levels. And what's more is when we look at these, this is GAD2 background, so with presence of ARMT9, and this is GAD2 ARMT9 double knockout with absence of ARMT9. With the ARMT9, the high and the low GABA accumulation, which cause different stomata conductance. However, in GAD2 ARMT9 double knockouts, the high and the low GABA doesn't affect the stomata conductance, which suggests when you have absence of ARMT9, then the GABA accumulation high or low would not affect the stomata opening. And take all this together, we here propose the model that compared to the wild type plants, the mutant GAD2 has very low GABA levels in leaves, and that actually maximized the anion uptake through the ARMT9 into the vacuole and deregulates the anion accumulation in the guard cell and makes the stomata constantly more open and drought sensitive. And compared to the normal condition, the drought stress increases GABA synthesis in the guard cell, and that inhibits the ARMT9 and further reduce the stomata opening during the day, help to save the water use, increase water use efficiency. And that story actually helped to backs up previous hypothesis stomata movement by the ADA. The ADA will help to close stomata, and the plant actually need another signal to prevent its reopening. So in summary, that GABA is a novel stomata behavior modifier in different plant species. It itself doesn't stimulate stomata change, but it can fine-tune the gas exchange, increase water use efficiency. And that's probably is through the negative regulation of ARMT9. And why the GABA inhibits stomata closure, actually, we're still working on that. I would like to thank all the people listening here, particularly my supervisor, Professor Matt Gilliam, and also thank the PhD student over here, the Charlotte and Shu-Ying. They did significant work to this project, and also our former postdoc Yulong and Professor Steve Tyman, and also the rest of the people listening here. Thanks for attention.
Thank you, Wesley. Any questions? And for those online, if you can type any questions in the Q&A box, please, and I'll read them out if you have any questions. No? So we have a question from Steve Timon. So stomata is still open with high GABA, and if it completely blocks ALMT9, what else is transporting malate?
Well, I guess there are some others, not just malates. There are some other anions to contribute to stomata opening. So, which means, first, it blocks the ALMT9, only partially impairs stomata opening, but not completely inhibits the opening. Second, there are other members catalyze the anion, such as CLC, what else? Yeah, such as some other members. And in fact, there are also some ALMT, like ALMT4, it's primed anion release from the vacuum. Hopefully that answers Steve's question.
Do you have any questions from the audience? Okay. Okay, if we don't have any more questions online either, I'd like to thank Zhou for his presentation. So thank you, everyone. And I'll get Okay, our next presenter is Zhou from Matt Denton's lab, and he's going to be speaking about increasing crop productivity by hologenome concept. So thank you.
Yeah, good afternoon. I will introduce my research topic, increasing crop productivity by hologenome concept. And today I will introduce a broader range of my research topics, and I won't focus on very specific research I'm doing. I hope to, set up more cooperation with everyone in the campus. In the past 30 or 50 years, we have a very good understanding about the plant genome. And we did a Sanger sequencing, next generation sequencing, and even now the nanopore sequencing. We have very, very good understanding about the plant genome, even the functions of the gene based on the one gene, one enzyme hypothesis. But plant genome cannot explain everything what we observed in the field condition. For example, the legume crop can grow without nitrogen in the soil, because the legume can use rhizobia to fix the nitrogen for the plant growth. So we look for the fixation gene, but fixation gene not in the plant genome, it's in the rhizobia genome. So are there any other genes the plant can use from the bacteria to improve its growth or adaptation? So when we consider the plant genome, we also need to consider the microbiome genome. So we call it a hologenome here. Yeah, the hologenome concept was suggested maybe 20 or 30 years ago. Yeah.
Sorry. Sorry. I want to get rid of it. It's being difficult. Sorry. Yeah, I'm trying to get rid of that. Come on. Sorry. Maybe we'll take. Okay, it's been difficult. Sorry. Sorry, we'll leave it.
No.
Yeah, sorry.
Okay. Yeah, the hologenome concept was suggested maybe 30 years ago, but why only recently this research become the hotspot? The problem is not the difficult to sequence the plant genome. The difficulty is from the microbiome genomes. And we can see that 1 gram soil is the main source for the plant microbiome. In 1 gram soil, there are around like 10 billion genomes, and 99% of these microbiomes are uncountable. So we can't see it. So how can we know their function and who are they? And recently, we developed the metagenomic sequencing and bioinformatics to understand the soil microbiome. There are 2 main approaches to do this work. One is sequencing a very specific gene, such as 16S. This can help us understand the taxonomy group of the soil microbiome, such as who are they. And another approach is the metagenomics, which sequences the whole genome, whole metagenome of the soil. This can tell us not only who are they, but also what their function. This is a very simple flow chart about the bioinformatics for metagenomic sequencing. Normally, we get the short reads from the sequencer, and we will do the assembly. We will assemble the short reads into the quantics, and then we convert the quantics into the genes. For the genes with similar sequence, and we will merge them into the non-redundant gene. Using this method, I did 1 single soil first. I sampled 1 single soil sample from the Roseworthy campus, and then I sequenced this soil metagenome as much as possible. As you can see, when I sequence more from this soil metagenome, the number of genes increased until the maximum, which means the second step is deep enough to cover most of the genes in this soil metagenome. Finally, I found around like 20 million genes here. This is a huge genome size compared with like the wheat genome, which is also a large genome crop. Wheat genome has all about like 0.1 million genes. Yeah, this only based on a single soil sample, and we can see that's many of genes. And now we want to see if any different soil sample can have more genes there. As we know that the soil property normally change very significantly. Even at 100 meters far in a paddock, the soil property may be very different. So we sample like 68 soil from the main cropping zones in Australia. We call it our Australian agricultural soil bank. And we sequenced around 3 TB data from these soil samples, around 200 wheat genome. Finally, we can clearly get like more than 3 million genes from our soil metagenome. So this gene catalog is the largest gene catalog in the world by now, based on a single study. So this can tell us, using the metagenomic approach, we can get the microbiome information very well. And now we want to use this microbiome information with the plant. I show you 2 examples here. The first one we want to see is drought adaptation of chickpea and its microbiome. In this experiment, we set up 4 factors. Firstly, we focus on the rhizosphere microbiome and the endosphere. Endosphere means the microbiome inside the root. And we sample the root from 2 different vertical niches. One is the root tip, we call it apical zone. Another way is the basal root, is the basal zone. And also we did the experiment in 5 different soil types. We want to see if the plant selectivity for microbiome can be affected by the soil, or it is a conserved colonization. And more importantly, we test 3 different chickpea cultivars. These 3 chickpea cultivars with different drought resistance. One, we call it drought resistance, DR, and another 2 is drought successful. drought resistant result based on 8 different environments in northern New South Wales. And the drought resistant cultivar is the most drought resistant one based on the long-term result. And we also understand the mechanism of this drought resistance one. They use a drought escape, which means it can grow fast, it can mature early, flower early to escape from the drought. And this is our result. For the rhizosphere microbiome, we can see the soil type driving the microbiome in the rhizosphere. You can see different colors mean different soil, and each color always cluster together. But if we go to see the endosphere microbiome, we can see our vertical niche determine the composition, such as the circle, which is the apical zone, they always get together, and then the basal zone together. The exception is the drought-resistant genotype. Its apical zone microbiome overlapped with its basal zone. In order to see this figure clearly, we calculate the similarity distance, the distance between apical zone and basal zone for each treatment. From here, we can see the endosphere. This is the rhizosphere. For this endosphere microbiome composition, we can see similarity between basal zone and apical zone for the DR genotype is much lower than the DS genotype. This means the high similarity between apical zone and the basal zone. And also we also calculate the diversity of the microbiome in different niches. We also found that the DR genotype has very high diversity in its apical zone and also the basal zone. So we see that for the DR genotype, which is the drought-resistant genotype, it can form a high diversity of microbiome in the apical zone of the root. And we want to know the reason why. So we focus on the root exudation. Why root exudation is important? We know that around 7% to 40% of photosynthetic carbon will be released from the root as the root exudation. Here we show that the apical zone of the root exude more root exudate in the basal zone. And also for the DR genotype, which is the drought-resistant genotype, it released more root exudation of the carboxylate amino acid, but less sugars. And for the specific compound, we identified these 3 compounds, which drought-resistant genotype released more than the DS genotype. So we think for the DR genotype, its root can exude more root exudate, and a specific compound form a more diverse microbiome at its root apical zone. And also here, it's about the core microbiome. What is the core microbia? Core microbia here we define as the microbia existing across all the different soil types, which means the plant can always select this microbiome in different soils. Here we can see that for the DR genotype, the endosphere, the apical zone and the basal zone share 3 microbias, and we can also identify who are they. So we think the DR genotype, it can specifically select these 3 bacteria, which related to its drought resistance. So I show you another study. This study focuses on the rhizosphere microbiome, but it's under a different cropping system. Firstly, we simulate different cropping system. We sample the intact soil core from the field, which with the root residue, declining root inside. We call it plus DR. The DR here is the declining root, different from the previous study. And the second treatment, we remove all the root residue. We only feed in the soil, use the soil to feed in the tube. We call it minus DR. In this two different simulated cropping systems. We grow wheat and chickpea, and we sample the rhizosphere of the crops to analyze the microbiome of the rhizosphere. And we can see this is under the plus DR condition, which means with the root, the decaying root. And this is the non-decaying root. And when the decaying root existing, and we can see the rhizosphere of wheat and chickpea share more microbiomes and functional genes, which means they have a more similar microbiome as the rhizosphere. But their rhizosphere microbiome are very different without decaying root. And also we sample the soil which attached on the decaying root, and then analyze the microbiome there to compare with the wheat and the chickpea rhizosphere under DR. And then we also see a large overlap here. So we set up a model for the rhizosphere microbiome formation under the no-tillage system. In the no-tillage system, there is a root residue in the soil, and it forms a detractosphere, which means the rhizosphere of the decaying root. And when the new root grows, follow the detractosphere, we will have an interaction, because the detractosphere microbiome are very active, and so their effect will overlap the rhizosphere effect and driving the microbiome in the system. And our findings also are also supported by the previous result. As this observation from White and Kirkegaard, we can see this is the root grow in the field condition. We can see that the crop root cannot fill the soil, but crop root follow the previous root channel. So we are thinking in the no-tillage system, maybe there is no rhizosphere, because all the rhizosphere is the previous crop's decaying root. Yeah, so I talk a lot about the metagenomics, and metagenomics can help us understand the total soil microbiome. But the problem is, how can we use it? How can we use it for the farmers? So this is the next thing I will talk about, and how can we use this Australian agricultural soil bank and the soil microbiome to improve the productivity for the farmers. And here we will focus on the cultivable microbiome. Yeah, the first study is about the diversity of chickpea rhizobia by my PhD student Ming. It's covered here. Yeah, I think chickpea rhizobia is a very, very good example or model to study the bacterial evolution in Australia. The first reason is the chickpea rhizobia for 40 years in Australia, we only have one commercial chickpea rhizobia strain. Secondly, chickpea rhizobia and mycorrhizobia and the chickpea have very specific symbiosis. And only chickpea rhizobia only inoculate chickpea. Because for some other legumes, the one rhizobia species can inoculate many different legume species, but this not happened for chickpea. And we, using our soil bank, we isolate around 80 different isolates. And we did a genetic analysis of this isolate. And we can see that this is the commercial strain here. We only have some strain in the same genetic group with the commercial strain. And we also see some other strains form different groups or different species. So this figure shows the diversification of the naturalized chickpea rhizobia in the Australian soil. And here we also test the infestation capacity of different rhizobia. And we can see this. This is the commercial one, and only very few isolates better than the commercial one. Most of them have the lower nitrogen fixation capacity compared with the commercial strain. But isolated strain has better adaptation, such as here for the pH. For the black bar here indicated where the commercial strain can tolerate a different range of the pH. We can see when the pH become low, the commercial strain cannot survive, but our isolates can. And also for the temperature, and our isolate can survive in the low and very high temperature condition. So after the long-term use of the same commercial strain, the isolate, the naturalized chickpea rhizobia, it has lower infestation capacity, but it has better adaptation to the environment. This is another study from another of my PhD student, Nasir. His research focused on G/G effect of chickpea hologenome. G/G here means rhizobia genotype by chickpea genotype. He used 5 chickpea cultivars with different rhizobias, and also one control is no rhizobia, but he used nitrogen fertilizer. Here we can see, if we don't use rhizobia, we just use nitrogen fertilizer, we can't see the difference of the plant growth. But when we remove the nitrogen fertilizer, but inoculate the chickpea with different rhizobia, the variation is huge. I think this can be a very good indicator for the breeding, maybe. Because in the breeding, if you only consider the chickpea genome, and you get the result like this, there's no difference. But if you consider the interactions between the rhizobia genotypes and the chickpea genotypes, the result may be very different. And this is the last thing. We focus on isolating the beneficial non-rhizobia bacteria from our soil bank. This research is done by our PhD student, Anthony. He isolated more than 700 beneficial bacteria. They call it a plant growth promoting bacteria, PGPR. And from these different bacteria species, Bacillus, Pseudomonas, Burkholderia, and also he did the mechanism research. This shows when we inoculate the beneficial bacteria, which is DPR, to the chickpea, it can promote the chickpea growth much more than the control, as you can see here. And also we can co-inoculate the beneficial bacteria plus the rhizobia, the chickpea. It's also increased the plant growth. It also increased the nodulation. So what's the next? The next, we will focus on the beneficial microbia for the wheat crop. As we know, the wheat crop is the biggest crop in the agricultural industry. We are hoping one day the farmer can inoculate the wheat with the beneficial microbia. Yeah, thank you.
Any questions for you, and any questions online as well? Yeah,
So very next one first. I suppose regarding methodology, I suppose the way to extract, isolate clean DNA from soil and is quite different than doing the cell cultures. So how do you adapt the protocol DNA extraction for every different type of soil in order to obtain clean DNA?
Yeah.
So the question is, how do you adapt your DNA extraction protocol for different soil types?
Yeah, first we use the commercial kit, parasoil DNA extraction kit. And yeah, that one working very well. And I think the question you mentioned, why we need to know why the DNA extraction not very well in the in different soils. I think the major problem is the humic acid from the soil. Humic acid from the like the tree, the organic degradation. That humic acid can bond the DNA, so it will affect the DNA extraction. But here we focus on the agricultural soil, which and also you can see from the agricultural soil collection, most of them from the dryland agricultural area, which has very low organic in the soil. So for me, we don't have any problem for the DNA extraction. But we had a problem before for some soil for different products. Some soil from the, like under the tree, they get very high humid acid content, and then we can't get high DNA product.
Lisa?
So when you, I think I missed the link. So when you say a drought-resistant chickpea,
yeah,
in the same soil as the drought susceptible, it's able to make associations with those bacteria that help it survive better under drought.
Yeah.
But if you put those bacteria and you inoculate a drought-susceptible chickpea, does that help the drought-susceptible chickpea, or?
Yes, but the thing is that we can't do that because some bacteria are uncountable. Here we use the metagenomic method, which means we sequence the whole microbiome. That way we know who are they. But if we want to use them, we want to isolate them.
Yeah.
Maybe impossible. Yeah.
Oh, okay.
Any more questions? Okay, if that's it for questions, thank you once again to both our speakers. And there's coffee and biscuits out the back with Lisa. Thank you.
Yeah, but let's check all at the end.